So it's currently recording.
Cool.
So, okay, so today we will be talking about our project for the semester, which is called Elucidating the Automatically Detected Features used by Deep Neural Networks for RFAM family classification by myself and Tom. And we will be doing this presentation. I'll briefly present the outline. The presentation will be split in two, where I will present the first results for binary classifiers and implementation details of our models, and then Tom will discuss the result for multi-class classifiers. During this presentation, I will briefly go over the purpose and background of our work, how we prepare the data for learning afterwards, and then some implementation details. Then I will jump into the results for the binary classifiers that we got, and then the discussion occurring after those results regarding sequence length, nucleotide and dinucleotide content, and longer interactions that will be able to be drawn from the results we have. So, as an introduction, so we were presented some results that indicated that our families could be derived from their sequences. So prediction of non-coding RNA families is achievable with machine learning following diverse sources that use feature vector generated from sequence alignments or neural networks directly from the primary structure. But in our work, we wanted to test if similar results could be achieved using only deep neural networks trained on sequence directly. So those results were lacking the literature, but we were confident that it could be achievable. So in that context, we proposed to verify first that these models can accurately predict RNA family directly from their sequence. Then we will use different strategies to tackle the black box interpretability problem of those models by exploring various input sequence paddings and shuffling of the nucleotide contents. We will discuss that later. It's like the ground of our work is exploring the interpretability of those models. And then finally, we will postulate on how these DNNs could be integrated to annotate new genomes. So for first, we were furnished with a data set of roughly 7,000 sequences from 24 families. So we had to first pad them to a fixed length because our model could not accommodate different lengths of sequences. So we had to, we used two different strategies. One is to pad with blank nucleotides, so or zero padded. And then we had to also test if it was still possible to train on with a random padding of sequences, so random nucleotides padded instead of blanks. And also we try to shuffle our data set. By shuffling, we mean that each sequence is internally shuffled and based on either nucleotide by nucleotide shuffle or dinucleotide shuffled. These was the two setups that we tried. We will discuss them later. And for targets, it was pretty simple. It was the R family. But we had two setups. One was binary, so R family or not. or for multi-class classification was to determine the R family, one of the 24 R families correctly. The implementation of the model was in Python using PyTorch language. We have our code available readily to be checked, but roughly, without entering into details, it's one hot encoding of the sequences, nucleotide sequences. Then we used either a small or a big network that we will discuss later. So depending on the number of nodes that we wanted to include. And of course, we used the L2 regularization parameter just to prevent overfitting. Then we use the binary cross entropy loss function for the binary setting with an NADeep optimizer. Evaluation of the model was done on a test set using 5-fold cross-validation, and we chose to evaluate the ROC-AUC metric to evaluate the performance of the models. So, as our first results, we will look into the binary classifier performances for the prediction of each of those 24 families. We chose to represent, so to have these families to represent a wide array of different sequence lengths. And also some of them are similar. So it's not a trivial task, but we see that our model were able to accurately predict in each of the in each of those binary settings to have a pretty good AUC performance for prediction. We will discuss how we can compare those results in the upcoming slides. Here on the right, we see also some training curves for each of those models that we show that they converge and they reach good accuracy on training set, and then reported on test set, they generalize good as well. So first thing we wanted to see if the DNN we were using was capturing, what part was the sequence length of those seeds playing for the prediction. So for that, we compared the results from the zero padded seeds that we gave as inputs and the randomly padded sequences with random nucleotides to imitate genome search, for example. So this would be a more realistic setup for R family classification. And we see that in that case, they both perform very well on test set, even though having their sequence length partially obfuscated by the addition of random padding sequences. So this was quite promising. That was saying to us, we can go ahead and use those models in a setting where it's not a fixed length or zeros, it's purely random at the extremities of the seed. Then we wanted to see if the model was only learning from nucleotide composition. So I mean, having a majority of A or G or T. So the nucleotide composition could play a major role in predicting, so for the DNN to predict. In that case, we were comparing the zero padded model and the zero padded model where the sequence seed was shuffled internally, so every nucleotide was given another position in the seed. So basically, you're losing all the dinucleotide and the long-range interactions that might be happening in the seed that could help us predict the structure, and we looked at the difference in performance in those settings, and we see that it still achieved a quite good performance. So it hinted to the fact that nucleotide content holds much information for the DNA to make a prediction, but still it achieved less good prediction performance. Also, we could also analyze those results in a different context, is when we randomly padded. So when we basically remove every information about the seed structure, when we randomly pad the sequences that were shuffled. So basically it's practically only noise that the model is learning. And in that case, it's still performing in some cases, but it's losing performance with that regard. But we will investigate that further. But it could, it kind of hinted to us that length and now nucleotide information composition is key to make prediction in that case, and that if you remove that information, then you wanted to check how the dinucleotide content and the longer interactions were playing to make prediction. So in this particular scenario, we force the model to select longer substructures of 3 nucleotides and more. And that might suggest there are strong indicators of that family and that identity. So in that setup, what we did is that we shuffled based on dinucleotide content. So we were keeping dinucleotide and nucleotide content within a family. And the task was to determine if the seed was a shuffle, so a fake seed of this family or a real seed of this family. And in that contest, we got very decreased performance in that case, meaning that the model for a family, it can use longer interactions, longer patterns to identify a family. Because when we remove it from a family, it's no longer able to predict very well. It's only based, so the prediction will be based solely on nucleotide and dinucleotide content, and length is removed. So length is removed from this experiment. So so I'm finished with that part, and I will let Tom continue with more.
Okay, so for the multi-class results, using the same data set of the 24 RFAM families, we have, we first needed to make some architectural changes to accommodate the multi-class setting. So the first thing is we changed the loss value. Instead of the binary cross-entropy loss, we're now using the negative log likelihood loss, which NL loss you can see here. And then also we substituted AUC for the most part for accuracy as our kind of main metric of performance, because AUC isn't very well defined in a multi-class case. You can still define it, and we do. We have figures for AUC, but it can only be done against 2 classes, so it's a 1 versus 1, so all possible pairs of the 24. It's the average of all of those individual AUCs between each of the possible pairs. Those are the AUCs that we use, that we report. And then we can see that they're plotted along here. And then another metric that we start plotting now is confusion matrices. So we can see here this big 24 class confusion matrix. And so this is for a not the highest scoring model that we came up with, but basically the same, indistinguishably different, like a few percent different. But we can see that this is using random padding and architecture of 100 neurons in each of the 2 hidden layers. And then another big structural change that we needed to make that we weren't really expecting was removing the L2 regularization parameter. Now, this is something that improved the performance in the binary classification case, but in the multi-class case, it's not. It actually makes performance much, much worse. So removing that was essential, and also training for 50 epochs. And we can see here along the diagonal, we have like a majority of the classes are correctly classified, and we can see that our accuracy levels correspond with what we would expect based on this confusion matrix. Cool. Next slide. Okay, so moving more into kind of what we were talking about before with the shuffling and kind of obfuscation methods for different nucleotide or different sequence information, we employed the same zero padding, random padding, nucleotide shuffle zero padding, and nucleotide shuffle random padding into this multi-class classification setting. And then also three different architecture sizes. So instead of being 5 and 5 for the small, and then 1,000 for big, we also have an intermediate size of 100 and 100. First off, we noticed that when we, unlike in the binary class, or in the binary case where 5 and 5 networks, so like the smallest network here, is able to, Leonard, can you see my cursor?
No.
Okay.
I think you can annotate.
Can I annotate?
So yeah, if you go just around this green thing, you can annotate, I think.
Can you see that?
Yep.
Okay. Okay, so we can see here that for the small case of 5 and 5 neurons in the two hidden layers, unlike in the other case or in the binary case where this can perform like decently well with high above 95 or above 0.95 AUC, here we have quite poor accuracy, very poor. It's still better than the expected random, but it's still very poor accuracy. But then expanding the architecture to be a little bit bigger, so 100 and 100, we get above 95% accuracy in the zero padding case. And then also moving forward from there, the results are in accordance with what we saw before. So when we move from the preserved sequences with just padding added, and we switch to actually shuffling the sequences, so that is removing it's still keeping the nucleotide composition, but it's removing the dinucleotide composition, and it's also removing longer sort of substructures. So when we remove those, we see that the performance drops dramatically. So and it's even more dramatic than in the other case with binary classification. So what's going on here is, in order to be able to distinguish between all of the different 24 different classes, in order to keep it all straight, the network needs to, for one, be big. It needs to be bigger than 5 neurons in each hidden layer to be able to manage all those different features. And it also needs to have the structure information. It needs to have more than just single nucleotide composition. It needs to have longer subsequences available to it to be able to make the good classifications based on those things. So yeah, next slide. So moving on to another data set to kind of try to experiment what we can with this system that we created. We wanted to classify different microRNA families. So we were provided with a data set, or another data set of 3 microRNA families with many more sequences than the RFAM sequences that we have from RFAM. So the RFAM seed sequences, which are the manually annotated RFAM sequences on RFAM for each of the families. So we tried to classify between the 3 different microRNA families that we had available to us, and we see that the performance is very, very good. With after a single epoch, the accuracy has reached 100%, as we can see here. So after a single epoch, 100% accuracy is reached. And then if we look at the confusion matrix, we can see that again, that 100% accuracy is made. And then this is, of course, in the validation phase. So this is generalization. This is quite good performance. So a couple reasons why this may be is comparison to the previous cases that we looked at before, because again, these are just separate RFAM families. So why should these perform so much better than the other case? And one of the reasons is we have so much more data for this data set. So here we only have 180, or in the other case, we only had about 7,400 sequences. Here we have over 180,000, which is many, like over 10 times more sequences, over 20 times more sequences. And the reason why the accuracy reaches its max so soon, and we don't see a curve kind of situation, we see more of like an immediate jump to 100% accuracy. Well, that's because since there's so much data in a single epoch, that the sequence, by the time it's seen every example, it's already seen so many examples that it can already generalize perfectly. That's a classification. And also, this is a little bit of an easier classification problem than the 24 class multi-class classification problem. Like there's a lot more going on in 24 classes than in 3 single classes. So that could be another reason why it's doing so well. So next slide. To kind of push this a little bit further and to kind of use some of the labels that we had in the data set, we just were wondering what we can use them for. We wanted to explore a target value that we were given along with that microRNA data set, and that is their numbers that correspond to the Drosha cleavage efficiencies for these microRNAs. So that's how efficiently, a metric, I'm not sure which metric, of how efficiently Drosha, the protein, can cleave these microRNAs. And we can see that it's known that that's based on structure, because obviously all protein and RNA interactions are based on structure. So we wanted to be able to use this value that we have. So we have these values for all the sequences. We want to be able to use that. So we, instead of building from the ground up a regression system, we sort of adapted our classification system sort of quickly and easily into what we call here a pseudo regression system, which is essentially where we take that target value and then we divide it into 4 separate classes based on the value of that number. So that cleavage efficiency. So we have the ones that are lower than -4, between -4 and -2, between -2 and 0, and then greater than 0. And so those are the 4 different classes, sort of classes we have. This is the first time that we're looking at a classification problem where every single sequence should be from the same family. So we expected it to be harder, and that is what we see here. So we see that the performance is quite modest. So if we look at, we also perform this for the various shuffling techniques as well, but we would expect 0 padding in accordance with everything else we've done to be the best at this, so to be the most effective. And that is what we see here. And we see that it's 51% accuracy. So that is much better than random, because random would be 25% accuracy. So we can see that based on the confusion matrix as well, that like the results are more clustered along the diagonal, but we don't see the same sort of high specificity that we saw in the last classification examples. This obviously is in part due to the fact that we're kind of bootstrapping this regression problem into a classification problem, and a big problem with that is you will have sequences that are very similar, but they'll lie like right on the border of 2 classes, and so the system may have a good understanding of that sequence's Drosha cleavage efficiency, but it won't really be able to predict it properly, because if it's off by a tiny amount, then it ends up in a different class, and that's and then it automatically gets the same loss as if it had predicted that one completely wrong. So basically, that could be a problem. But we do see that
into the confusion of this classification problem, it's confusing the model in some
Yeah,
way.
yeah, yeah. Yeah, so it's confusing the model because very similar sequences, again, will have what it sees as very different labels, because there are only two labels. They're either identical or completely different, and it will see two very similar sequences as being completely different, and it has a very hard time learning from that, as we can see by the very sporadic training curves that we can see in the loss function.
And
Yeah,
also
so
we can also ask ourselves if this value of this cleavage efficiency is solely predictable from the sequence. That we don't
that's
know.
true. We don't really know much about it. Yeah.
But still, that we have signal for those models, even in that very grossly put setting, it's encouraging that some information is encoded in those sequences, in those microRNA sequences.
Yeah, exactly. So there's some signal. Yeah, there's some biological signal. It could be that this is a very difficult problem, or that, yeah, that it's an impossible problem, but we know that there's some sort of biological signal that reflects. So in a generative case, or in a generalized case, it is able to predict with some effectiveness the cleavage efficiencies, as we can see. But of course, in order to fully get an understanding of how well this can be predicted, this value can be predicted from sequence, we need to implement a full regression system. So next slide. Oh, also, I suppose I should just mention really quick that we see the same sort of progression and degradation of accuracy as we go, as we lose sequence information. So as we lose, going from zero padding to random padding, we lose length. Going from random padding to shuffled sequences, we lose longer than we lose longer than nucleotide composition. So we lose dinucleotide composition and longer structures. So when we lose that information, we do lose a lot of accuracy, which makes a lot of sense. That whatever the system is picking up on in these sequences, it's a sort of a structure-based thing. It's not length. It's not nucleotide composition. It is structure. So next slide.
And so just to go back. So this is performing less than random. So basically, we're saying that this model has not trained at all, or it has some kind of noise. So this is useful to keep in mind.
Yeah.
And also the AUC is like practically random, 0.5 being random.
So in conclusion, we tested different implementations of these simple feed-forward deep neural networks in the context of RNA classification. So we used sequences, raw sequences, one-hot encoded as input without using any structural processing step, no alignments or anything, just raw sequences. And we use different sequence shuffling schemes to determine which structural features are important. So nucleotide composition, sequence length, and different things like that. What are being detected and what are being learned by the neural network. And we show that we're able to get good binary results and good multi-class results in classification setting, and we're able to explain a little bit of where that results come from. So what kind of features are the neural networks detecting to be able to make their classifications. And we hope that this work will reinforce the usage of these DNNs as sort of a data-driven approach to non-coding RNA annotation by capturing key features that are encoded in RNA primary structure and accurately predicting their family. So we hope that these will someday be used on whole genome annotation type systems. Next slide. So quick future work. First thing to do is to benchmark these systems, so sort of adapt them to that, what we were talking about, the non-coding RNA and genome annotation situations, and then also compare them to state-of-the-art systems for this task. So Infernal, Structurinator, and others. So kind of compare them and see where this system falls. And also further experiments for the pseudo-regression sort of system is converting that system directly to a classification or moving from pseudo regression, that we need to fix that. Moving from pseudo regression to a full-blown regression system where we can predict real valued numbers for these Drosha cleavage efficiencies to see how well this system can perform. And also understanding sort of in the worst case scenario, which we showed for that Drosha cleavage, the pseudo regression task, determining the difference in the performance between the shuffled zero padding and the shuffled random padding, which appeared to be almost random, as if the system was learning about on just simple noise. We could be able to kind of elucidate what exactly is going on there, if it's really not learning anything. Next slide.
Ask a random question, so that we have maybe a Q&A at the end. Like,
okay, sure.
It's stupid, but I think it could be useful. Do you think that, so obviously predicting cleavage efficiency for microRNAs is different from predicting RNA family, but cleavage efficiency is linked to microRNA potency as target for treatment and so on. So do you see those models being useful in that kind of setting?
I mean, I would suppose so. Well, it all comes down to sort of what it's detecting on a sequence level. So if it's detecting the sort of structures that are important, and not just detecting some kind of other feature that we didn't think of or we didn't notice, then that might be confusing if you tried to do actual biochemical experiments on these microRNAs. But if it really is attending to these sort of structures, then maybe, yeah, that would be important.
So basically we have to investigate further if we're practically learning something from the structure with those experiments, or because if not, in that case, do you think that it would hinder the potency of those models, those predictive models for microRNA efficiency for some, because those microRNAs, they're becoming more and more used.
Yeah, for sure. I mean, maybe an example of some way that might be like that we might not understand for this is maybe it's not protecting or maybe it's not predicting like the Drosha actual binding site. Maybe it's predicting something else that's heavily linked to that. So it's not really detecting the structures that actually bind to it. So if you were to use this intuition to design different other experiments based on this, you might get unexpected results if it's not, if it's detecting something that, some sort of modification or something like that.
But we have to go back, and with previous methods, we always have to predict some kind of structure based on an expert system that will say, well, this sequence will yield this structure, this structural feature. That is, it is known that this feature will bind to this structure. Now we have proved that maybe it might be discovered from data itself.
Yeah.
And we saw that prediction goes up when we accumulate more and more data. So
for sure,
it was quite striking to see that for the microRNA experiments, since we have a lot more sequences, the model is performing very easily.
Yeah, for sure.
And so with the R families that we had, those 7,000, does it cover all non-coding RNAs, or?
No, it's just like a few sequences from various sources that were on RFAM. So it's like, I mean, there's tRNAs in there. There's a couple different kinds of tRNAs. There's ribosomes, like 16S subunits and like stuff like that. But yeah, and then there's like a couple microRNAs and like a couple tmRNAs. So there's a few.
But all those seeds were annotated by experts. They weren't predictions of our families. We were sure about their families.
Yeah. Yeah, they were the
Pretty
seed.
simple.
That's the seed. That's why they're called the seed sequences, because they're used as seeds for the prediction algorithms, which is exactly what we did.
So basically, we were recreating some kind of annotation system.
Yeah, if we ran our predictor over a bunch of genomes and then got a bunch of potential hits, that's essentially what RFAM is. It's just a bunch of seed sequences and then a bunch of potential aligned sequences. Yeah.
So basically, in our work, we recreated the RFAM system, but in much smaller it's like a miniature of
Yeah.
the RFAM. And
Yeah.
we didn't even learn. So what is funny about our work is that we never looked into the structures themselves. It's
No, yeah.
purely
That was
sequence and machine learning.
that would be difficult. Yeah, it would be difficult to look at the sequences or to look at the structures. But yeah. So, do you have any idea how long how long that was?
So this is why I ask questions, because we were really quick.
Oh, okay.
So I will now stop recording.